Hi guys, welcome to the first in a series of lectures on classical genetics. Classical genetics can be compared with modern genetics or molecular genetics in that it simply deals with how traits are inherited. If you'll recollect, at the beginning of the year, I taught you guys about 7 common characteristics of living things, and one of those characteristics is heredity. Heredity is the ability of organisms to pass traits from parents to offspring via DNA. And so remember, we've just spent quite a bit of time studying DNA, and we found that in the DNA molecule in humans, we have about 21,500 different genes. And of course, genes are codes for proteins, so we make 21,500 different proteins. A slippery concept in genetics is for me and for lots of students in the past, has been this idea of an allele. So I'll keep coming back to it, but in short, you have a gene for eye color. You have 2 different copies of that gene. One copy could code for a blue eye color protein. One copy of that gene might code for a brown eye protein. And we say these are 2 alleles for the eye color gene. Here he is, Gregor Mendel. He's the father of modern or classical genetics. He was an Austrian monk. He lived in a monastery. Remember where he slept, if you remember Robert Hooke's observations. And his work went largely unnoticed for 100 years, I think, before it came to light. And what he did there in his monastery was he taught biology, which is a very exciting profession. And he also grew pea plants. Now, he studied those pea plants, but I'm sure he ate a lot of peas as well. And one of the things that he employed was a technique called selective breeding. And so in selective breeding, you take pollen from one plant and you germinate or you fertilize ova from other plants. And so Mendel spent a lot of time breeding plants. Of course, humans have been selectively breeding organisms for tens of thousands of years. Our common domesticated animals like the pig or the bull or the horse or the dog are all the products of selective breeding. Of course, probably the first organisms that we selectively bred were plants, like wheat. So one of the common misconceptions is that plants reproduce asexually. That's not true. Plants reproduce sexually. And remember that sexual reproduction is all about the production of male and female sex cells or gametes. And in order for you to sexually reproduce, 2 gametes come together to form a whole new organism, as opposed to asexual reproduction in bacteria. Well, there's a couple different ways that a plant can be fertilized, and those 2 are self-pollination and cross-pollination. Notice that in a flowering structure, or a flowering plant, we have both the male and the female reproductive organs in the same place. And so the male organ is called the stamen, the female organ is called the pistil. And on the stamen are those male cells called pollen. In the interior of the stigma or the pistil is an ovary where there are lots of ova that can be fertilized by that pollen. Now, pollen needs to get down through the stigma into the ovary, and that can happen a couple ways. One way is for the pollen from that plant to be blown by the wind or carried by a pollinator to the stigma. And another possibility, of course, is that pollen from other plants nearby travels to the stigma and fertilizes the eggs. So self-pollination versus cross-pollination. What Mendel did was that he took pollen from a plant that he desired to cross and delivered it to his other selectively bred plant. And he did this with a paintbrush and some scissors, so it's a relatively simple process. And what he ended up with was true breeding plants. So when you selectively breed, a lot of time what your aim will be is to pick specific characteristics and breed for those characteristics until you get a strain that doesn't or always demonstrates that characteristic. And we call these pure or true breeding strains. For example, a German Shepherd. And the key to a true breeding strain is this. If you take a German Shepherd that's purebred and you mate it with a German Shepherd that's purebred, you'll always get German Shepherds. It's kind of a gray area, but people spend a lot of time breeding animals and plants. There's all kinds of strains of roses and strains of chickens and strains of sheep, all with cool names. Which one's your favorite? How about here? Is it the ram cat, the pocket horse, milk cow, or the Easter bunny? Well, Mendel selectively bred pea plants, and he selectively bred for 7 different characteristics. And those characteristics were the position of the flower, the color of the flower, how tall the plant was, the shape of pods, the shape of peas, the color of peas, and the color of pods. And what he did was he selectively bred these plants for these characteristics until he found two traits. And so, and for the characteristic of flower position, there were 2 traits. The flower position could be axial or terminal, that is on the end or on the axis of the plant. For the characteristic flower color, he bred 2 true breeding strains for the traits purple flowers or white flowers, etc. So he did that for these 7 different characteristics. He had 2 strains of true breeding plants for each of those characteristics. And then he did his experiments. Here's some genetics language here. Note that Mendel took 2 true breeding strains of pea plants, Pisum sativum, and the true breeding strains were either purple or white. And when he bred those 2 together, those parental generations, which we denote as P, he ended up getting the F1 generation, or the first filial generation. And there was something odd here. He took a purple plant and a white plant, bred them together, got all purple. And that doesn't make any sense. The question is, where did the white go? And this question, this observation that he made, allowed him to test and try to formulate a model to explain what he saw. So Mendel came up with some new terms here. He said that when you bred 2 true breeding strains, and one of the characteristics or one of the traits disappeared in the F1 generation, he said that the trait that was expressed means seen, was dominant. And the trait that had disappeared, he called recessive. So the next experiment that he conducted was the F1 self-cross. So he took those F1 generation plants and he bred them together. Remember, they were the product of a purple and white plant, and they were all purple. But when he bred those 2 together, he got an F2 generation, the second filial generation. And he noticed something odd. White had reappeared, but it reappeared in a strange ratio. In fact, he did this same series of experiments for those 7 characteristics. He crossed yellow pea plants with green pea plants. In the F1 generation, he got all yellow plants. When he took those F1 yellow plants and bred them together, he saw green reappear, but in a roughly 3-to-1 ratio. So that's the observation. For each of these traits, he said, he saw the 3-to-1 ratio in the F2 generation, and it allowed him to formulate a simple model for the inheritance of traits.